And so it's a pleasure to be back here in Concepción. So I gave a talk 3 years ago, I think, in marine genomics course. And actually at that time we were much more focused on cell biology, clocks, and now that I'm part of the LOMIC, so Laboratory of Microbial Oceanography, we really want now to promote our model organism to address environment question. And I think that another title would be, this is ultimately what we would like to do, including here in Chile. I'm going to talk today mainly about anthropogenic effect and about the use of biosensor. But another title that would be very appropriate is how can we use model organism, here I'm going to talk about Ostrococcus, to address environmental questions. And this has a very big potential. We recently had a workshop in Banyuls, which was called When Genetics Meets Oceanography, and where we could see that people are going toward this direction. I would like really to give you insight into this kind of novel approaches. So my talk would be split in 3 main parts. The first one has to do with the presentation of Ostrococcus, that some of you probably know, some not. Most of the talk would have to do with the development and the use of luminescent reporter line to assess mainly the toxicity of biocides, pesticide, but also oil contamination, and a study that we have recently initiated in collaboration with Rodrigo de la Iglesias in Santiago about copper toxicity, still using the same kind of luminescent bioreporter. This, I would say, this is the main, so far, the main interest of our group. I should say that this anthropogenic effect is more like a side project that became really big, because when we started developing this luminescent bioreporter in the lab, we found that there were many, many people interested. And so from a side project, it's becoming almost a main project. And and that's actually very nice to see that people can use our tools to address other questions. But our main interest is what's called the phenology, and so it's a very strange thing, but our phytoplanktonic blooms. In a more simple word, what we are interested in is determining how the environmental forcing, mainly by light and temperature, and also, of course, nutriments, affect the development, the proliferation of and division of phytoplankton cells, so which leads to the formation of spring blooms, and this mainly in pico-eukaryotes. Okay, this is our favorite alga. It seems huge, huh? But actually the size is 1 micron. So it was described as the smallest free living organism. Actually, it's here. There was a paper published by Claude Curtis, who was in the lab. He just retired. So Ostrococcus was discovered only about 20 years ago in the Tow Lagoon, and at that time was described as the smallest free living eukaryote. So it was discovered by flow cytometry, because at that time what Claude saw by flow cytometry is that there was a very small population that didn't look like cyanobacteria because it didn't have the right pigment, but was looking more like other photosynthetic organism. And in collaboration with Marie-Jo Dinet, they get nice images showing that actually this is a eukaryotic cell with a nucleus and chloroplast, a mitochondria, starch granule here, which is out, and actually is one of the smallest organisms known today. This alga is able to grow very quickly in the lab. We can get 2 to 3 generations. It's very easy to cultivate, and more importantly, in my group, we have developed all the genetic tools so that we can analyze biological function. We can create reporter, luminescence reporter, to follow biological processes. One example is, for example, recently we have generated ferritin mutant. So the ferritin is an iron storage protein. To determine the role of ferritin in iron storage, and in the response of cells to iron limitation. So this is just an example, but you can do that, of course, for many, many different processes. And this is very powerful to be able to invalidate a function so that to determine the importance of this function in adaptation to specific ecological niche. So of course, what I didn't say, the genome of Ostrococcus was sequenced several years ago. It's very small, very compact. For genetic approaches, it's very, and to invalidate a biological process, it's very convenient because there is very little gene redundancy. It means that if, for example, there is one ferritin gene, not 5 or 6, so if you do a knockout in the ferritin gene, that you know that this cell is not going to be able to store iron anymore. So, and so we have all these genetic transformation tools, including gene replacement by homologous combination. And this is, I'm not going to go into detail, but for eukaryotic cells, this is really very, very powerful cell because you can do precisely what you want to invalidate a gene or to express a gene somewhere in the genome. Okay. So Ostrococcus was initially found in the Thau Lagoon near Montpellier. And at that time, there were several people who say, okay, looks interesting, it's very small, but this is something that is just in the Thau Lagoon near Montpellier, and who cares? Since then, actually, Ostrococcus has been found in many different places. It's a blooming species. Of course, it belongs to prasinophytes and has the small cousins, like Micromonas, and Bathycoccus. You probably know about these cells because they are found everywhere. This Ostrococcus, for example, blooming species, it's found along all the American coast, including in Chile, in the upwelling system. There was a paper published by the group of Ed DeLong recently showing that in metagenomic data, actually, Ostrococcus is one of the most abundant phytoplankton cells along the Californian coast in this cruise. So it means that these are not diatoms, but are still important ecological function. Micromonas, for example, is found to be dominant in phytoplankton in Arctic. So it makes sense to study all these small pico-eukaryotic cells. So now I'm going to move to really the main topic of my talk, is that the development of luminescent bioreporter to assess toxicity and to report, I mean, more generally, the expression of genes in vivo. That's really the important point, is in vivo. It means that you can follow gene expression over a very long time, as you will see in the following slides. So this is, what I'm going to talk is about a study that we did in collaboration with Fabien Jou in Banyuls. And was published last year in IOM. And so we use Ostrococcus luminescence line as biosensors to evaluate the toxicity of pesticides. Also, just to give the general background, so pesticides and biocides in general can be quite abundant in coastal areas, especially on the Mediterranean coast, and it's even more important in the lagoon, because the lagoon are closed environment with lots of human activity. So it comes from, of course, sorry, I'm trying to find back the pointer. Okay, it's back. So agriculture activity, so it's mainly herbicides, can be also neurotoxic. So herbicide, you have a diuron, which is not allowed anymore. But also biocides, which are used as antifouling, and some compounds like diurons, which are not allowed anymore as herbicide, are allowed as biocides. So you don't get them from now from the river, but you get them directly from the boat in the harbor. And actually, the concentration of these biocides can be quite high in coastal areas. So especially, as I say, because Mediterranean region can receive usually a little rain, but when it rains a lot, and they are characterized by big flood events, and that can wash the ground and bring a very large amount of these concentrated pollutants in sensitive area in this lagoon. And it's also very important in this lagoon because there is lots of aquaculture in the lagoon. So this molecule, of course, can affect the activity, viability of lots of marine organisms, including phytoplankton. And when you are talking about herbicide, of course, you think mainly about photosynthetic organisms, means that phytoplanktonic species. So there are different kind of biocides that I mean, they used to be like TBT. It was very toxic. It was banned in 2003. Copper can also be used as a biocide, and actually, I think it's used in salmon farm for the cage. Two here common biocides are diuron and ergarol, and there have been several reports in the Mediterranean Sea showing that you can have concentration up to 1.7 microgram per liter of diuron and 0.6 of ergarols in harbor. So this is a locally very high concentration. So now, if you want to test the toxicity of biocides, there are different ways of doing it. I would say the ideal way, the most realistic, is to do mesocosm experiment, because really you are working on community. But this is a big problem, because if we want to do, statistic with triplicate and everything, and if you want to do dose response curve, you can imagine how many of these guys you will need to put on your beach, and even with that, you will have very few time points. So this is ultimately what you want to see, but this is not very easy to perform. Other things are approaches consist of using model species that are recognized to monitor toxicity. So you can test many different conditions, especially if you are working in microplates. You do many replicates. Of course, this is much much cheaper. Inconvenience is that it can be less realistic, of course. And of course, there is also another very important issue. In Europe now, there is a directive which is called the REACH directive, according to the name of the guy who did this directive, because now there is a large diversity of molecules and of their photoproducts which are issued from the degradation, and for lots of them, we do not have many ecological tests, or we have, but only on some organisms. It's really important to investigate on a larger scales about the toxicity, the potential toxicity of this molecule. And if you do that, it's very important to have a very high, large, high throughput screen on many different types of organisms. So what we have chosen to develop is the luciferase reporter line. So this is very schematic, but it explains well what we do. We take a gene of Ostrococcus, of course. To these genes, we fuse the luciferase of firefly. So the fireflies are naturally able to emit light, and this is made by hydrolyzing luciferin. This is a substrate that you add to the cells, and it produces oxyluciferin and photons. And with a luminometer, just a photomultiplicator, you can directly measure the light emission, which is proportional to the expression level of the gene. And this is non-invasive method. It means that you can measure the same sample for a very long time. For some genes, we have been measuring for over a week the same sample every hour. So if you want to do dose response curve, it's really very, very convenient. So this is to illustrate how we generate this line. It's not very simple. This is something that we developed in my group. It took us several years to do that, and we are still improving it. So we start from culture. We resuspend them, we spin them down, resuspend them in sugar solution, because then what we do is electroporation, so apply electric field. And of course, if you do that in seawater, it just makes big bang, and nothing enters into the cell, so you have to move them to sugar solution. Applies your field, then you can give them different construct with a selection gene, and then different construct, either just a promoter of the gene fused to luciferase, or the full gene, means the promoter plus the protein fused to luciferase. Then we select the transformant which are resistant to the antibiotics, which is carrying the vector, to select the transformant. And then we can grow them and start playing with them. We also have very efficient protocol to cryopreserve them so that we can keep them for several years in -80 freezer without any problem. So then once we have this line, you are 96 wells, and you can of course do all your concentration dilution of pesticide, test different pesticides. I mean, on the plate you have 96 possibilities. Initially, we started just with 1 luminometer and start monitoring toxicity. But then, since you know, when you have people in your lab, they tell you, oh, that's nice, but that's not enough. So we want another luminometer. Then you buy a second luminometer, and then they come to you and say, it would be nice, we are many people, if we could get a third luminometer. And you say, okay, we can, but even if you have money, then you have a problem of space, because you say 1, 2, 3, 4, and after, you know, a few years, you will have your lab full of luminometer and no space to put anything else. So then we say, okay, now I told them we are going to stop, because there are other ways of doing it, and so we just kept our best luminometer and took a robot. So we have a robot now. We can handle 6 plates, which is the equivalent of 6 luminometers. And of course, because we play with photosynthetic cells, we want to be able to give them some light. So we have also designed a special setup so that we can give them light from underneath, and then the robot will just take the plate, move it to the luminometer every hour, and do a measurement, and that over the 6 plates. And since our main interest, I will talk about that at the end of the talk, has to do with all the day-night cycles, the rhythm, the light quality, then we add small LEDs here that can give all kind of light curve, blue, red, green here, solar curve, whatever, I mean, all the shape you want. And then all of this, the cells are monitored every hour and goes to a database. So with the kind of thing that not what you want to do for biosensor, but that's what we do. We want to monitor circadian rhythms. This is a clock gene, for example. You can monitor many, many different cells, and here you see very nice oscillation of genes that under circadian control. This is just, you know, this is not mathematics, this is biology. And it shows you that you measure precisely something with the right tool. Biology is highly, highly reproducible. And this is just 1 microplate. Actually, not even, is like 8 wells. So you can do that for the 96. So now we move back to the main objective of this talk is, can we use this kind of reporter to assess biocide toxicity? So what do we do at the beginning of experiment? We had pesticide, I think here it's tyrone, so you have a control, and then and then you monitor automatically every hour. So there is nothing to do, the robot does that for you. And so students like very much this experiment, usually because they start the experiment, and then Carlos, who is here, actually has been doing that with copper, start the experiment, then they go, they play on the computer, and they listen to music, and the next day they come back, and they say, oh, it's finished, now we can work. And so it's very convenient. And so then you can normalize, of course, and look at here as the inhibition, calculate your EC50, and here there are just 5 curves, but for every well you can have 96 measurements. Since when we started this project, as I say, it was not our main project, it was just a side project. And I had discussion with Fabien in Banyuls, and we were both convinced that it was really nice to try this kind of approach on Ostrococcus. But we didn't want to start by designing specific line because it take a lot of time. So we just took all the constructs that we had published for different stories, clock genes, cell division genes, and we to look at the effect of pesticide. Okay, and so what we found here is that if we look here at the different line, we found that the best one actually was the CDKA, because we got a 50% inhibition after 32 hours. For the other one construct, it was usually much longer. Or this one is usually quite low level of expression, so it's not as easy to use. So now, of course, when you do this kind of experiment, you need to validate, because this is just luminescence. So, and the classical way of doing toxicology study is to follow cell growth. In our case, we do it by flow cytometry. At that time, it was a Beckman cytometer. Now we do the same with an Accuray. And you can just count your cells. And when you do that, students usually don't like that very much, because they cannot stay on the computer, play with game, because they have to sample, you know, every 2 or 3 hours, fix, and in the end, you know, they get curves that don't look very nice. So, I mean, they really prefer the other thing. You can ask Carlos, huh? So, yeah, this is the advantage and disadvantage of both assay. Of course, luminescence is non-invasive. The other one, you need to sample. It means that if you want to do those response curve, is also a problem in a 96 well if you need to take 20 microliter to do your inhibition. This is high speed, of course, because it takes 8 minutes per microplate, whereas 2 hours, even when you have a microplate injector in the flow cytometer. And the other thing, since you are always measuring the same thing, you have low variation, and so very small standard deviation in this assay compared to this one. We did this for, so the best biosensor we found was a CDKA biosensor. So with this CDKA, we have tested lots of different molecules. We found some EC50, 0.75 for ergarols, or 5 for diurons, which were quite similar, I mean, in the range of those that the concentration that you can detect in some harbors. So it means that maybe we thought that we could use this bioassay not only to monitor the toxicity of the molecule, but also maybe to test this on environmental sample, directly seawater samples. That's because this is really the ultimate goal. So now if you compare here with the literature, so this Ostrococcus CDKA reporter line, so EC55 after 48 hours. If you compare that to growth inhibition on different species, it can vary between 0.5 and 45. So some are a bit more sensitive, most of them are less. But after 72 hours, if you take after 48, usually it's actually more. And compared to the classical, I would say, luminescent test, which can also be automated, the microtox or in Vibrio, or here in the quick light in dinoflagellate, or reporter line in cyanobacteria on Escherichia coli, you see that here you have in the range of 1,000, and here you have really 4 ranges of magnitudes below. So it means that for this type of molecule, our sensor is quite interesting. Of course, when you do this kind of studies, you want to know whether this is realistic with respect to what you can see in the field. So Fabien did some experiment where he looked at the effect of diuron. Here, various concentration between 0.25 and 25. So in the range of what could be found here in some harbor. And look at the effect on chlorophyll, and after 5 days here, and you see that you can have a strong effect, and the same here for ergarol. So then he focused more on this in detail, and by flow cytometry on the different community, cyanobacteria, pico-eukaryotes from Banyuls, so which have also Ostrococcus, and nano-eukaryotes. And what was interesting is that what we found that the most sensitive community were pico-eukaryotes here, because you see that you have a much stronger inhibition here, and for the 2 pesticides that we have used, actually. So it means that Ostrococcus, which is a pico-eukaryote, is a representative of one of the most sensitive group, at least for this molecule. this is actually an interesting small story that we also did with Fabien recently, because we say, okay, it looks like this bioassay is working for pesticide. What about other molecule? And an obvious candidate was oil, because as Jeff said, we like very much oil in the lab, so we have to put it everywhere. And of course, if you can think about spill, there was this deep horizon spill in April 2010 in the Gulf of Mexico, the BP platform with more than 800,000 cubic meter of oil that were discharged, and more than 8,000 cubic meter of the dispersant corexit that was used. This was very convenient, this dispersant, because there was lots of oil. You add dispersant and just it's gone, it disappears. So all people are very happy because they can go back to the beach and swim and everything. It looks like everything is back to normal. But that's what we wanted to look at using our reporter. So the question is oil, can we detect some toxicity for oil? What about the mixed oil dispersant? Is it less toxic, more toxic for phytoplankton? So we did some extraction because oil is not soluble, and the components were measured: benzene, toluene, hexanes, and also some extraction in the presence of the dispersant at 0.08%. For this, we have so far we have not had the composition of what we extract in the soluble fraction, and we apply to the algae because we apply only the soluble fraction. This is the kind of thing that we can observe. Again, inhibition, an EC50 after 25,000 ppm of oil, which is rather high. So it means that oil is not very toxic on its own for Ostrococcus. Then, if we look at the dispersant plus oil, you see that it's much more toxic at 2,000 ppm, which was surprising that the mixed oil dispersant is very toxic. And there is a 12.5 factor. So then we look at the effect of the dispersant alone, and we found that actually this dispersant alone, the corexit, is highly toxic to the cell at 0.6 ppm. So it means that the dispersant on its own is much more toxic than the oil, and the dispersant on its own is much more toxic than the oil. And then when you mix oil with dispersant, the oil reduces the toxicity of the dispersant, which is quite amazing. So it means that it was maybe not the best idea. I mean, of course, if you want to clean the beach, but you use this dispersant because it has a very strong impact on phytoplankton, at least on Ostrococcus. Just two slides also to show what we have been doing with Carlos. We came to Banyuls at the end of last year. So it has to do with the copper toxicity. So this is a collaboration with Rodrigo in Santiago. So they have been working for many years now on the contamination in the channel area to study copper toxicity. And here you have the channel where the water coming from the mine is discharged into the sea, and where you can have up to 4 micromolar of total dissolved copper, which is quite high. So the idea again is, okay, we have used this for herbicide, pesticide. We have used the biosensor for oil. What about copper? Because every time you show this talk, then people say, okay, this is nice. Maybe we could test that on copper, or maybe we could test that on salmon farming. So, I mean, we're happy because people seem to like this kind of approach. Then, from this study, there were several conclusions. Since that time, we had been generating different reporters. So, I mean, the CDKA is a classical one here, for which we could calculate an EC50, which was close to 6 micromolar, so it's quite high. We also use another reporter, the ferritin, because we are working quite a lot on iron in the lab, and since we suspected there could be some interaction between iron and copper metabolism, and indeed what we found is that the EC50 for this ferritin luciferase reporter is much lower. Is about 3 micromolar. This also tells you that there is just not one reporter, that there are really possibilities there are 8,000 genes in Ostracoccus, and by finding the right gene, you can have some reporters that are more sensitive. So the EC50 is about 3.5 micromolar, which is still quite high. And so the last experiment we did with Carlos was to test the effect of water from the riverine by doing different mix after adjusting salinity with seawater, with non-contaminated and contaminated water. And we found an EC50 which is close to 2.3 micromolar. And so that's interesting when you compare the EC50, because there are lots of other things in this river, and the pH is lower. There are lots of other things, other metals and components. It means that by just comparing the EC50, what this tells you is probably that most of the toxicity that is coming from this river is due to copper, not to other things. At least copper in culture can account for most of the toxicity that we see in the natural sample. So there are many advantages of using this luminescent biosensor. It's highly sensitive, reproducible, it's really cheap. What it costs, of course, time and money to develop the line. But once you have it, you can run, especially with automated things, hundreds of samples in parallel. We have sensitivity which is similar to what, at least for Diron and Irgarod, what we see on natural phytoplanktonic community in Banyuls. So very quick and possibility of automation. So what are the perspective? As I say, all the genes are not responding equally to contaminants. So what we are trying to do now is to select some biosensors that are more specific, not just take all the light that we have been using in the lab that are luminescent, but try to get more of them, more specific. And to do that, we have started to do, that's also something we do in collaboration with Carlos and Rodrigo, to do some experiment where we add different concentration of copper to the cells, and then we do RNA-seq analysis, and the idea is to identify some genes that are induced very early and at very low doses in response to copper. So that we could, and the idea, of course, is try to, if it works, this kind of approach to extrapolate. The other thing, of course, is now to try to use this on natural samples, because this is the ultimate goal, and that's something that we have started to do in collaboration with Jeff, and this is very, very preliminary, but it looks like Ostracoccus luminescent light could be used to detect some toxicity in natural samples. That's in the Vermeil Ecotox project. And even more interesting, I would say, that in these samples, it looks lots of chemicals and pesticides have been measured. In some of these samples where there is very high toxicity, we do not detect anything known. This means that this could be a discovery pipeline to identify molecules that were not supposed to be toxic but are very toxic for phytoplankton. Okay. I don't know how much time is left. Oh, okay, so then the last point that we are interested in, of course, because this is the main part of my project. I could give another talk if you like. So it has to do with what is important in determining the timing of phytoplanktonic blooms, and that's why we are here also, because I think several people have very similar interest. So, I mean, the classical hypothesis, squared-up hypothesis, was done, I mean, in the 50s, is that what triggers the phytoplanktonic blooms is that after episodes of water mixing during the winter, when the temperature rises, then you have water stratification, which remove light limitation, and then when you have both nutriment and light, then you can initiate the bloom. But I mean, this is generally probably true, but now there are several examples that suggest that it's more complex than that. There are a combination of physical, chemical parameters, bottom-up and top-down. Also, there is some predation, interaction with bacteria, and very importantly, probably interaction between light and temperature parameters. So this is so this is a project that we have that was almost financed last year, but we missed for just one place, and we hope it's going to be financed this year by the ANR in collaboration with people from Montpellier. The Tour Lagoon, of course, is very interesting for us because Ostrococcus tori is very abundant there, and we can play with our favorite model. But also the Banyuls Bay, where there have been monitoring for several years, and you see here in green that every year, about January here, you have a very nice peak of pico-eukaryotes. Now, if you compare the Tour Lagoon and Banyuls, it's interesting because we have two different situations. In Banyuls, you have the spring which precedes the temperature augmentation, whereas in Tour is the opposite. You get first the temperature, and then you get increase in nano and pico-eukaryote communities. So it means that depending on the place, you may have some different, forcing that can initiate the bloom. Temperature and light are very interesting. They are also very interesting to us because in the context of global warming, we know that temperature is increasing. Light, at least the day length, is not going to change because it's just linked to Earth's rotation, and coupling both forcing, of course, may have some very important effect on the development of these communities. Then I come to the point that is our main interest. We have been working on that for many years. So the question we can ask, is there photoperiodism in phytoplankton, like in terrestrial organisms, that where now we can compare the trees with the algae, not only with the coral. So what is known in plants, but also in animals, called seasonality, is that the time of many events is regulated by the day length. It's what's called photoperiodism, I mean the time of flowering and growth in plants, for example. And so this photoperiodism is arise mainly from the coincidence between the external time, the day length, and our internal time, which is given by an internal clock, the circadian clock. And this is very well illustrated in plants. For example, when you have clock mutants, what it happens is that instead of flowering at a certain time of the year, they will flower at another time. And this have very big impact on the development, and one of the examples is for a plant that will normally flower in summer. In a mutant, for example, you will have flowering in the spring. And of course, this is very, in terms of ecology, can have very big impact. So the question is that, are there clocks in phytoplankton? That's what we have been mainly studied for almost 10 years now. So you see here, you have by flow cytometry in Ostrococcus culture. That was before we had luminescence, so we are going with students, and me too, going to the lab and doing time point every 3 hours for 5 days. But in the end, you get a very nice curve here, very periodic increase. And more interestingly, you see that when you maintain these cells under constant light, you still have this periodic increase. And this is typical of circadian rhythm. So we have circadian rhythm in Ostrococcus, and there are some other reports showing that there are circadian rhythm in phytoplankton. So if you have circadian rhythm, you have a circadian clock, you can think that you have potentially photoperiodism, like in other.
What is the y-axis in the previous one?
Which one?
The y-axis is 80 to 180.
This is, well, it depends. I mean, actually, there should be, here there should be two. Here in yellow is the number of cells, but there, which is probably this one, and this is a FL3 parameter, chlorophyll. Yeah. And so what happens is when the cell grows, the chloroplast increases, and the time of division, then here, the splits are divided, the cells are split in two, and then the fluorescence per cell decreases. So that's a nicer way of saying it, but it shows the same thing. So this is just, and here it shows, this is just the number of cells here, and where you see this periodic increase when the cells were untrained for different day lengths. Okay. So is there a clock in Ostrococcus? Yes. To make short a long story, we identify clock genes which are similar to clock genes, which are using our luciferase reporter. We show that they have expression pattern consistent with the clock. We show that when we do mutant, then we lose the rhythmicity. So we clearly have clock genes, and now we have clock mutants. And the very interesting thing also that we show is that these clock mutants have phenotypes. It means that, for example, we have a mutant which is smaller, and so actually we have the size mutant of Ostrococcus. And that's funny because Ostrococcus was described in the initial Nature paper in 2014 as the smallest free living eukaryote. So now we can say that we have a mutant that is smallest than the smallest free living eukaryote. But that's not, I mean, that's just, but what it shows that this clock is very important to regulate the cell size, the biomass, and of course with respect to photoperiodism, potentially, it may have a very big importance, this circadian rhythm. This is just to finish what we want to do now, where we are going. We have been doing lots of cell biology, playing with mutants. We are now playing with biosensor, with environmental samples. We want to do experiment now not only on mutants, but also microcosm experiment on pre-bloom communities, so to test the effect of light, temperature, and see how they can affect the bloom. Of course, we want to still to investigate that in our mutants. And even add mutants in microcosm experiment in the lab, because now we have also fluorescent mutants that we can follow by flow cytometry and compare that to other natural population. And this is just to show lots of people. I don't have picture of everybody. So Fabien for the biosensor part is really leading. We are starting collaboration now with many more people, Jeff and Cesar, Philippe, and engineers, Jean-Claude, and also Valérie help engineers lab who develop all the techniques of transformation, generate the construct. Hugo is a PhD student. He works a bit with Carlos. So Carlos who came to do the work in copper in collaboration with Rodrigo. And Fanny and Sophie, we work also with Fabien and us for the first story on the pesticides. And that's all. Thank you.